So it's my pleasure to introduce Jesse Bloom. Jesse Bloom he did his PhD and postdoc at the California Institute of Technology, and since 2011 has or been a professor at the Fred Hutch Cancer Center, as well as an affiliate member of Genome Sciences. And a couple of years ago, he also became an investigator of the Howard Hughes Medical Institute and has been doing very important work on SARS-CoV-2 evolution this year. So without further ado, we'll hear from Jesse.
Hi, so thanks everyone for coming and listening to my talk today. So I'm going to be talking about the evolution of SARS-CoV-2. So first of all, I'm going to put the link to my talk here in the chat. So hopefully you see that in the chat, and you're welcome to go to that link if you want a copy of these slides or to follow along in any way. And then I'd also just like to encourage people to feel free to interrupt and ask questions during the talk rather than waiting to the end. Okay. So hopefully you can see my slides. If you can't see my slides right now, someone should unmute themselves and tell me, but otherwise I'm going to get going on the talk. So my talk is Interpreting the Evolution of SARS-CoV-2. And so since I'm talking about the evolution of this virus, I want to begin by taking a step back and pointing out that for most of the history of evolutionary biology, people studied evolution just by looking at how things changed in terms of their phenotype. So this is a famous image from Ernst Haeckel, who was sort of an early biologist. It's called The Pedigree of Man. And in this picture, he's arranged all these different species in this tree-like structure. And he's done this just based on sort of looking at how similar they appear. You know, and a man is more similar to a gorilla, so he puts them close together in the tree. And so this is how people studied evolutionary biology for most of the history of the field. But about 55 years ago, it also became possible to study evolution in another way, which was in terms of changes in genotype. So I think the first paper of this type was by Emil Zuckerkandl and the famous chemist Linus Pauling at Caltech. And what they were doing is they were working at a time when it had just become possible to start to determine the sequences of proteins from different species. So they had the sequences of myoglobin proteins from a variety of different species, like horses and humans and chickens. And these sequences are strings of letters. So if we were looking at DNA sequences, they would just be A, C, G, and T. But these are protein sequences, which was what they were able to determine at the time. So there's actually 20 different letters, and they're just each of these sequences. And so what Zuckerkandl and Pauling did is they took these sequences and they arranged them in a tree-like structure, sort of based on how similar the sequences were to each other. And at the time, in 1965, this idea was very intensely criticized by a lot of really famous evolutionary biologists as sort of being a trivial and nonsensical approach. The idea being, what could you possibly learn about the interesting, characteristics of a horse and a human, or a chicken and a tuna, just by looking at these changes in these protein sequences? The idea was that was too trivial to tell you about the phenotypes we really cared about. And Zuckerkandl and Pauling recognized this, and they wrote that how the change in a gene sequence, so its genotype, how it affects its phenotype, so how it actually sort of works in the real world, is really complicated. And in particular, they wrote, there's no reason to expect that the extent of functional change in a protein is proportional to the number of amino acid substitutions. They said it's the type rather than the number of changes in the protein that happen that's decisive. And so this was sort of this transition point in evolutionary biology about a half century ago. And where we stand now is that people have almost forgotten that we ever used to study evolution by doing anything other than looking at genes and genomes. So the field of biology has gotten very good at sequencing things. And so we can now characterize evolution of genotype at really high resolution. So for instance, it's possible for anybody to go to websites on the internet and get phylogenetic trees that have over 1 million SARS-CoV-2 sequences. And these sequences are arranged just based on how similar the viruses are in their sequences. So you can basically see which viruses are related to which other viruses. And you can see this in really exquisite detail. It's possible, for instance, to track exactly how the virus is mutating, which viruses are descended from which other viruses, and so on. However, often what we still want to know is not how just the sequences or genotypes of these viruses are changing, but how those changes in sequence or genotype actually affect the phenotypes that we might care about. And so here's an article on this topic that came out at the end of last year, which is called "The Coronavirus is Mutating. What does that mean for us?" And what I hope you can appreciate is what the New York Times is asking, which is, what do these mutations or change in genotype sort of mean, or what do they imply for a change in phenotype, is really the same question that evolutionary biologists have been worrying about for a long time now, which is, how does the number of mutations or substitutions you can see in a gene or a genome, which is a really easy thing to kind of measure and count up, relate to sort of the type of change that we might care about in the real world. And so this is sort of the topic that my lab studies. We're really interested in, for proteins and genotypes from viruses, how changing the sequence of part of a virus will change the sort of things about the virus that are relevant for the virus out there in the real world. So sort of giving meaning to all these genotypic changes. And so that's what I'm going to talk about today in the context of SARS coronavirus 2. And so my talk is going to go like this. First, I'm just going to explain to you what is the SARS coronavirus 2 spike and what is the receptor binding domain, or RBD, which is just a small part of this virus, but turns out to be very important. Then I'm going to just talk a little bit about what we know about how coronavirus spike proteins evolve. Then I'm going to tell you how we can actually determine how specific mutations will affect the spike protein and receptor binding domain of SARS-CoV-2, and then what all of this means for antibody recognition and antibody immunity against the virus. Okay, so first, just some background. So if you were to look with really high magnification at a SARS-CoV-2 virion, you would see something that looks sort of like this. So here's one virus, and on the surface of the virus is this protein called spike, shown here in red. And there's a whole bunch of copies of this protein that are sort of sticking out of the surface of the virus. And here's actually the molecular structure of this protein. This is exactly what it looks like. And this structure was actually determined by David Wiesler, who's a scientist at University of Washington early in the SARS-CoV-2 pandemic. And so the reason that SARS-CoV-2 virions have these spike proteins on their surface is it's because these are the proteins that allow the virus to get into a cell. And so a really important part of this spike protein is a region that I've colored here in purple, called the receptor binding domain, or RBD, as I'm going to refer to it. And so the spike is just one part of SARS-CoV-2, and then the receptor binding domain is just a relatively small part of spike. But as we'll see, this part turns out to be really important for a lot of the phenotypes that we care about. And that's because the function of the receptor binding domain is to bind with really high affinity to a protein called ACE2. And ACE2 is a protein that's expressed on the surface of some cells, including some human cells. And sort of the binding of the SARS-CoV-2 spike's receptor binding domain to ACE2 is the first step in the virus getting into a cell and initiating infection. So this is a super important process. And the part of the reason that SARS-CoV-2 was able to cause a pandemic is because it has a receptor binding domain that can bind really well to human ACE2. In addition, we know that neutralizing antibodies, so these are the antibodies that are able to directly block viral infection, and these are the types of antibodies that are elicited, for instance, by a SARS-CoV-2 vaccine. These neutralizing antibodies always bind to the spike, because that's the thing on the surface of the virus that it uses to get into cells. And they often bind to this specific part of the spike the receptor binding domain. And in many cases, the way they work is they just sort of bind to it and get in the way with its ability to bind to ACE2 and just sort of block that whole process. So you can imagine then that mutations in the spike, and particularly the spike's receptor binding domain, that might change how well an antibody can bind there and block infection could be really important. And in fact, we'll see in a little bit that they do turn out to be really important. Okay, so hopefully that's kind of explained to you what I mean when I talk about spike and the receptor binding domain of SARS-CoV-2. And now, before I want to get into the details of how SARS-CoV-2 is actually evolving, I just want to talk at a high level about how coronavirus spike proteins evolve. And so a really important thing to note when you're studying viruses, which is sort of an example of the general principles of evolution and genotype and phenotype that I was talking about at the beginning of the talk, is that although all viruses evolve in the sense that their sequences are changing, only sometimes do these changes in sequence lead to large changes in antigenic phenotype. So two examples of this that probably everyone's familiar with are two different respiratory viruses. So measles virus and influenza virus are both viruses that transmit by the airborne route. So they're both respiratory viruses. And furthermore, both measles virus and influenza virus have genomes that are made out of RNA, like SARS-CoV-2 does. And viruses that have their genomes made out of RNA rather than DNA tend to have very high mutation rates. So both these viruses have very high mutation rates. Nonetheless, these viruses are very different in terms of what we would call their change in antigenic phenotype. So their change in the ability to be recognized by the immune system. So measles virus does not evolve to escape immunity. So prior to the advent of vaccines, people got infected with measles virus at most once, and usually exactly once during their lifetimes. And then in the 1960s, a vaccine was developed against measles, and that vaccine still works really well today. If you take that vaccine, you're nearly guaranteed not to get infected with measles. In contrast, influenza virus evolves rapidly to escape immunity. So people are infected, they make an immune response, they make antibodies against the virus, but then they can be infected again about 5 years later with the descendants of that virus. And the reason is every year influenza is changing in a way that helps it get away from the antibodies that were elicited by the prior infection. And as a result, the influenza vaccine has to be updated annually to try to chase after the virus's evolution. So one of the questions that we wanted to ask when SARS-CoV-2 first emerged is, how would SARS-CoV-2 behave? Would it be like measles virus, or would it be like influenza virus? And at the time, and still to some degree today, you can't totally answer that question just by looking at SARS-CoV-2, because there isn't a lot of sort of past behavior or past history of SARS-CoV-2 to study. So what we decided to do is we decided to instead study some of the relatives of SARS coronavirus 2. So SARS coronavirus 2 is not the first or only coronavirus to infect humans. There's actually 4 what are called common cold coronaviruses that have been circulating in humans, for a decent amount of time now. And those viruses actually cause about 30% of all common colds, but they haven't historically been studied very much because they only cause common colds. They don't cause severe disease. So in general, most scientists didn't think they were worth studying. And one of those common cold coronaviruses is called coronavirus 229E. And this virus causes common colds and has been circulating in humans since at least the 1960s. And in fact, it's probably been circulating in humans for much, much longer than the 1960s. That's just when it was first identified. So we decided to study coronavirus 229E, the idea being that since this thing is related to SARS-CoV-2, maybe if we understand how coronavirus 229E evolves, we'll get some insight into how SARS-CoV-2 might evolve. And so this picture has a lot of data on it, but what I just wanted to convey the point is we reconstructed the history of how the spike of coronavirus 229E has changed over the last, about 4 decades. So this is what's called the phylogenetic tree. The horizontal direction here is time. So things over on the left are older. You can see the viruses up here have dates like 1979, 1982, 1982, 1984, etc. And then the viruses further to the right here are the newer viruses. So you can see these viruses are like from 2016, 2019, etc. And so you can see this particular virus evolves in such a way that the population is always sort of fairly closely clustered at any given point in time, but is sort of changing in a more or less synchronized way over time. So there's not a high, if you're an evolutionary biologist, you would say that this virus has low standing diversity, but rapid change over time. And what that means is if you just sort of identify the time, so where you are on this horizontal direction, that tells you pretty well where you are on this tree showing how all the viruses are related to each other. So we just picked viruses from 8 different time points at 8-year spans across this tree. So 1984, 1992, 2001, 2008, 2016. And we made viruses with those spikes in the lab so we could study them experimentally. And then we just did a really simple experiment, which is called a neutralization assay. So what you can do is you can take serum, which is the liquid component of blood, essentially, and that's where the antibodies are found. And you can ask, how well can the antibodies in that serum block a virus's ability to infect cells? And the ability of serum to do that is called the neutralization titer. So a high neutralization titer means the serum is very good at blocking the infection. The technical definition is the neutralization titer is the reciprocal of the dilution at which the antibodies can still block the infection. So for point of reference, if you've gotten a SARS-CoV-2 mRNA vaccine, like the Moderna or Pfizer vaccine, you will have a neutralization titer to SARS-CoV-2 that's maybe a little bit higher than 300.
Hey, Jesse.
Mm-hmm.
You mentioned that people should interrupt with questions. There's a few in the chat that you probably can't see them.
Yeah, I'm probably not
one
reading
of them,
it.
One of them is a clarifying question about, do you mean you synthesized reconstructed ancestors? I thought you might want to clarify.
Yeah. So that's a great question. So we didn't so when we went here, we didn't actually have to computationally infer the ancestor because we directly knew it. So they weren't inferred ancestors, but they were just, the sequence that someone had literally directly sequenced in 1984. We just synthesized that gene. So it's not the inferred ancestor, it's like the actual historical sequence. But yeah, that's what we did. And let me see, let me figure out how I find the chat. Are there other questions that I've been missing here? Or yeah, if there's other questions, just feel free to, I don't think I'm gonna be able to find the chat one.
Yeah, that's okay. The other ones are more general. You can do at the end.
Okay, sounds good. Oh yeah, the slides are, well, I'll still go these. The slides they're HTML slides, so you can view them, but I don't think it's a good way to download them. Yeah, and the other ones I might wait to answer because they are kind of more general. So sorry, here, now let me close the chat. So yeah, just interrupt if you have questions, or Billy, you can feel free to interrupt. Okay, so basically what we did is first in this experiment, we took serum that had been collected in 1985 from someone who was 26 years old back in 1985. And since people are infected with these common cold coronaviruses, you would expect that this person in 1985 had been infected with a virus pretty similar to the ones that were circulating then. And indeed, you can see that this person's serum can neutralize the virus from 1984 really well. So it's really potent against that virus, like almost as good as the immunity you get from a SARS-CoV-2 vaccine. But then if you sort of look into the future from 1984, 1985, you see that by 1992, the virus's spike has changed enough that the serum is able to neutralize it much less well. This neutralization titer has gone down almost tenfold over those 8 years. And then if you look, 9 more years in the future, it's gone down even more. And when you get to 2008, so about 25 years later, this person's serum, which worked really well against the coronaviruses 229E that were around then, basically doesn't have any neutralizing activity against those sort of future viruses. So this is showing that coronavirus 229E behaves more similarly to influenza virus, where you get this drop due to viral evolution, and less similarly to measles virus, where once you have immunity to one measles virus, you're immune to all the rest of the measles viruses you could be exposed to for the rest of your life. So basically, we do expect coronaviruses to change in a way that will erode antibody immunity. Nonetheless, because people are always worried about this, like what's the virus evolution going to do to their vaccine? It's really important, and I try to use the word erode rather than escape. As you can see that the sort of the units on this x-axis are years, right? In fact, a lot of years. So it's taking a number of years for the virus to change enough to erode this immunity. It's not like, you know, your immunity works today and then tomorrow viruses change so much that it's not working. So it's a gradual process, but definitely as the virus evolves over time, it's eroding that immunity. And then just one other interesting point I want to make out is that although the virus changes in such a way that it's always eroding people's immunity, because different people are heterogeneous and have heterogeneous specificities of their immune responses, the erosion happens at different rates for different people. So for instance, this individual here starts with a neutralizing titer of a little over 1 to 100, and after 8 years, the virus's evolution has totally eroded that antibody neutralization. On the other hand, this individual over here on the right starts with about the same neutralization titer, a little bit over 100, but then their antibody immunity stands up really well to the viral evolution, and it's going down over time, but it's going down really slowly. And we think that's probably because the antibodies that this person made happen to bind to a different part of the virus than the antibodies that this person made. And the antibodies that the person on the right here made are probably binding to a part of the virus which is changing more slowly. So we're now trying to understand these types of differences in a lot more detail, because you can imagine if you were trying to design a coronavirus vaccine, ideally it would elicit antibodies like the one here on the right, where it may take decades of viral evolution to really erode that immunity, rather than antibodies like the one here on the left, where, you know, after 8 years, it might be totally eroded. And so we don't know the answer to that yet, but we think that by studying these historical things, we might learn some ways about sort of the best way for the immune system to recognize a virus in order to stand up to viral evolution. Okay, so I now hope I've told you how sort of coronavirus spikes evolve and what these spikes are. And now I want to get to the really specific question of how would the exact mutations affect the function of the SARS-CoV-2 receptor binding domain? And the reason I want to get to that is what I just told you, you know, understanding the general principle of how coronavirus spikes evolve is important for understanding what we think will happen next. But what we'd like to be able to do as scientists is not only know that this virus is going to change in such a way that it will eventually erode immunity, but we'd like to know exactly which mutations are going to do that. So when people are doing all this sequencing of these viruses and a new mutation pops up, we can understand whether that mutation might be important or not. And so in particular, for the SARS coronavirus 2 receptor binding domain, we would like to prospectively map how all of the mutations affect the relevant phenotypes. And I want to emphasize we're just measuring specific sort of biochemical phenotypes here in the lab. So a challenge when you study a virus is that there's something called the fitness of a virus. And that's really basically a measure of how well the virus transmits in the human population. So for instance, many of you may have heard of the Delta variant, which is this new emerging SARS-CoV-2 variant, and it's better at transmitting in the human population than the viruses that came before it. So the Delta variant's been spreading very rapidly across the world. But that fitness is, you know, a property of how the virus transmits in the human population. And that's not something you can measure in the lab. Like, we can do a lot of things experimentally, but we can't measure viral transmission on humans in the lab for a variety of logistical and ethical reasons. But what we can do is we can measure sort of the biochemical properties of a virus's protein that we think contribute to its fitness. And so for the receptor binding domain of the SARS-CoV-2 spike, which, as I told you, is this really important part of the virus, we think there are 3 main biochemical phenotypes that contribute to fitness. First, this protein has to fold into the particular structure that it uses to perform its function. Then it has to actually perform that function. which is bind to ACE2 with high affinity. And then those are the two things it needs to do to get the virus into cells. But you can imagine when people have antibodies, there's a third thing that the receptor binding domain might sort of want to do or benefit by doing, which is changing its ability to be recognized by antibodies against the virus. So approximately speaking, we think that the evolution sort of operates under the following constraints. The virus has to stay good at these two things because those are important to be able to infect cells. And meanwhile, it wants to change this third thing sort of as much as possible. So it's affected as little as possible by the antibodies that people are making after vaccination or prior infection. So how are we going to measure all these biochemical phenotypes? So in fact, we're able to do a lot of this using really sort of fairly simple lab systems. In fact, we use yeast, which many of you may have heard a lot about in Matreya's talk last week at Wednesday in the Genome. And so yeast are just the super convenient sort of model organism in the lab, and we can take yeast and we can just put the receptor binding domain on their surface. So make the yeast express the receptor binding domain on their surface. And then we can use a fluorescent tag to see if we've gotten a receptor binding domain on the surface. And we can incubate the yeast with something like an antibody or a fluorescently labeled ACE2 to see how well the receptor binding domain binds to ACE2 or is bound by an antibody. And then there's lots of techniques we can use to separate yeast cells based on these fluorescent signals and sort of use that to measure these phenotypes. And then the really important thing that we do is we don't just do that for sort of the current SARS-CoV-2 receptor binding domain. We make a whole bunch of these yeast, each of which have a different mutant of the receptor binding domain on their surface. So in fact, we have all of the possible single amino acid mutants of this receptor binding domain on the surface of this big pool of yeast that we just do for our experiments. And so this allows us to generate a whole bunch of data. So I'm going to show you what that data looks like, and I'll try to explain it in a sensible way. So don't freak out on this heat map too much. I'll explain what it means. So as I was describing, there are two biochemical phenotypes that we think are sort of represent the function of this protein. One is that the protein has to bind to ACE2 with high affinity. And the second one is that sort of in order for it to even have a chance of binding to ACE2, you've got to express the properly folded protein in the first place. And so this is measuring how each mutation affects these two phenotypes. So there's a whole bunch of positions in this protein. They start at 331 and go on up to about 200 higher than that. So all the way over here. Each amino acid, each site can be any one of the 20 amino acids. So for instance, right here, this is the particular mutation changing site 350 from a V to an E. And so I've colored these mutations red if they're bad for that property, white if they have no effect on that property, and blue if they're beneficial for that property. So if you just sort of look through these maps, you can see there's a lot of red because many mutations are deleterious. Mutations are usually bad for a virus. But there's also a lot of white because there's a lot of positions where mutations don't do much of anything. And then occasionally, there are positions where there's blue. So occasionally mutations actually help these properties. And so, if you don't really care much about the SARS coronavirus 2 receptor binding domain, that's probably all you need to know. But right now, there's also a lot of interest in really understanding what the specific mutations that arise mean. So for instance, in late 2020, there were new SARS-CoV-2 viruses that are now called the alpha variant, or at the time were called the B.1.1.7 variant, or just the UK variant, that had a mutation called N501Y. So we could go to our map and we could look up the N501Y mutation, which I've highlighted right here. And you can see that mutation allows the virus to bind to ACE2 better than it did before. And so that might actually aid in viral transmission. And that could be one of the reasons why this alpha variant did quite well and spread to a large degree through the world. You can look at other mutations, like some of you may remember that there was a whole lot of excitement about SARS-CoV-2 viruses infecting mink farms in late 2019 and early 2020. And the reason for that is when these viruses infected mink, they often got this mutation called Y453F. And when you look in this map, you can see that that mutation is another mutation that enables the virus to bind to human ACE2 better than it did before. So basically, there's all this data here. It's on this website. Any of you or anybody else who wants can go look it up. And the nice thing is that when people are sequencing viruses and finding mutations, they can then just go to this map and look up sort of what that mutation does. So it's sort of a useful way for interpreting the effects of these mutations. Okay, but the thing I really want to focus on is, so people are interested in the function of this protein, but the thing that people are most interested in when they see and hear about these mutations is how do the mutations affect the ability of antibodies to recognize the receptor binding domain. And so when I showed you our little yeast system, I talked about how we can sort of incubate the yeast with anything we want that binds to the receptor binding domain. And so one thing that binds to the receptor binding domain are antibodies. And so now I'm going to show you how mutations affect binding by an antibody. So first, I'm going to talk about a specific antibody. This is a monoclonal antibody called LY-CoV555 or bamlanivimab. So this is an antibody that was developed by Eli Lilly as a treatment for SARS-CoV-2. And so, okay, so here is the three-dimensional structure of the SARS coronavirus 2 receptor binding domain, shown here in gray, and this antibody, bamlanivimab, that was developed by Eli Lilly, shown here in pink. As you can see, the antibody binds to the receptor binding domain, and that's how it's able to block infection. And then what we did in our experiments is we went and we looked at the mutations at every position in the SARS-CoV-2 receptor binding domain. And we said, which of those mutations allow the virus to escape binding by the antibody? So most of these mutations are down here on the x-axis because they have no effect. But you can see there are a few positions like site 452 or site 484 or 472 or these other positions right here where mutations cause escape. So they're up from the x-axis. And in fact, you can see all of those mutations are right where the antibody binds to the SARS-CoV-2 receptor binding domain. So that's why they're able to knock off binding of that antibody. And so this information is really useful to have because this was an antibody that was being rolled out to treat people who were infected with SARS-CoV-2 prior to the ability for people to get vaccines. And an unfortunate thing about this particular antibody is that it's affected by mutations like at site 484, which turns out to be a site that's mutated in, for instance, the beta and gamma variants of SARS-CoV-2, which are pretty common, and also the Delta variant of SARS-CoV-2, which has become extremely common. So this antibody, even though Eli Lilly put a whole lot of effort and money into designing it, basically was obsolete almost as soon as it was approved by the FDA because the virus had mutations like this one and this one, which escaped antibody binding. Fortunately, some other antibodies, which I'll go to in a little bit, for instance, the ones by Regeneron, turned out to do much better against the mutations that have shown up so far. And so I think one thing that this indicates and that we're kind of excited about is going forward when there are new viruses, when people or companies sort of choose antibodies to develop, they should look at more than whether just does the antibody bind to the virus well, which this antibody does. They should also look at, are there mutations that are likely well tolerated by the virus that can knock off the antibody? And perhaps if that had been done more prospectively, for instance, Eli Lilly could have chosen an antibody that wouldn't have been degraded so quickly by viral evolution. Okay, so that is what happens, how mutations affect binding by this one specific antibody that was developed by Eli Lilly. And like I said, we've now done this for a lot of antibodies. So here's another little website we've created where there's this huge list of antibodies here, and you can kind of go and look and see how each of them are affected by mutations. So here's LY-CoV555, or the antibody I was just talking about, and you can see it's affected by mutations at positions like 452 and 484. And here are all the antibodies arranged by how similar they are. And there are a lot of antibodies that are sort of similar to this one and are also been very affected by these mutations. But there are other antibodies. So for instance, S309 here is an antibody that was developed by Vir Biotechnology. And this particular antibody, the mutations that affect binding by this antibody are at positions down here, which are not positions that have really been mutated much in SARS-CoV-2. And you can also see that this is somewhat of an unusual antibody. When we sort of group all the antibodies together, this one's pretty far away from most of the other antibodies. And so this is sort of a unique antibody that's probably a pretty good antibody. And increasingly, companies are trying to find antibodies like that one. Or here's another one that's being developed by Vir, S2H97, which also has a pretty unique escape profile and isn't affected by common mutations. And hopefully these monoclonal antibodies will be much more durable treatments because they'll be less likely to be evaded by these viral mutations. But we think the other thing that's just nice is for all of these antibodies out there, when a mutation shows up, someone can now just go, and say, all right, this particular Regeneron antibody, REGN 10933, what are the mutations that are going to affect that? Is it going to be impacted by a mutation in some of these variants? So this is useful phenotypic information to have to interpret the virus's evolution. However, fortunately, at this point, most of us are not relying on monoclonal antibodies anymore to protect us against SARS-CoV-2. So just to sort of make the distinction, a monoclonal antibody is a defined antibody sequence, which is identified and then developed by a company and used as a treatment. So there are monoclonal antibodies that are used to treat cancer, and there's monoclonal antibodies like the ones I'm just describing that are used to treat SARS-CoV-2 infected individuals. And those antibodies sort of bind to one defined place on the virus. And if the virus gets a mutation where the antibody binds, the antibody usually doesn't work anymore. However, the amazing thing about our immune systems is that when you're infected or vaccinated against the virus, your immune system makes a whole bunch of different antibodies. And so these are called polyclonal antibodies. And these antibodies can potentially bind to lots of different parts of the virus. And so that makes the polyclonal antibody response much more resistant to viral evolution. Because you can imagine if the virus gets a mutation that knocks off this purple antibody, the gray and blue and green ones are still binding. So there's still antibody immunity against the virus. However, sort of how broadly this polyclonal antibody response actually binds across a virus can vary from person to person and virus to virus. In some cases, maybe it just will happen to turn out that a lot of the antibodies are all binding to a similar region rather than this sort of broad binding that I've drawn here. And in fact, we know that the extent of focusing of these polyclonal antibodies is a big factor in shaping how a virus is able to evolve. So for influenza virus, if you take the polyclonal antibodies in a person's serum, you find that they're often highly focused in which particular part of the virus they recognize. And so it's not uncommon for just one mutation to influenza virus to be able to reduce the ability of a person's antibodies to neutralize that virus by over an order of magnitude. In contrast, for measles virus, these polyclonal antibodies tend to broadly target many epitopes with similar potency so that no single mutation has a large effect. And this is probably one of the reasons that measles virus is not very successful at evading human antibody immunity. Okay, so obviously, what about SARS-CoV-2? So we've also sort of done this mapping I was just showing you for polyclonal antibodies. So what I'm going to do here is I'm first going to show you what this looks like for convalescent serum. So this is serum that's elicited, the serum antibodies that are elicited by infection. So first of all, if you just look, first I showed you all these different monoclonal antibodies. So these are individual antibodies that people have isolated. And you can see that the polyclonal antibody response, so the net effect of, sort of all the antibodies in someone's serum is sort of typically in the middle of where all these monoclonal antibodies find, which makes sense because it's kind of the average of it. It's usually closest to these purple ones though. Anyway, so if you look at that, you can see for the polyclonal antibody response, there are certain positions such as at site 484 and 456, and so on, where mutations can strongly reduce binding by polyclonal antibodies. So there are some pretty big peaks here. Importantly, those peaks aren't the same for everybody. So some people, like this person, has a very focused polyclonal antibody response. Mutations at site 484 have a really big effect. In contrast, that say this person, mutations at site 484 don't have such a big effect, but this person would be more affected by mutations to the virus at positions like 446. And then there are some individuals where mutations at no sites have much of an effect. So anyway, this has been useful to understand. It turns out 484, which tends to be the most strongly targeted site, is actually a site that's mutated in some of these SARS-CoV-2 variants, like for instance, the beta and the gamma variants. So the virus is getting mutations that are starting to erode these polyclonal antibodies. But again, as I emphasized, these polyclonal antibodies can bind to many parts of the virus. So the virus is going to need multiple mutations to escape from them all. And so one thing that we've been working on doing is using knowledge about where these polyclonal antibodies bind to figure out what combinations of mutations would start to really erode the antibody immunity. So work by structural biologists like David Wiesler at University of Washington and Pamela Bjorkman at Caltech has sort of defined several different important regions, which here we've conveniently called the class 1 region, the class 2 region, and the class 3 region, where the potent antibodies tend to bind. So you can imagine to really erode this antibody immunity, the virus would need to get a mutation where the class 1 antibodies bind. It would need to get a mutation where the class 2 antibodies bind and where the class 3 antibodies bind. And so virus is going to need combinations of mutations to get rid of, to sort of get away from all of these antibodies. Unfortunately, there's no viral variants out there that really have mutations in all of these important epitopes right now. But again, we're kind of trying to work on calculating what that would look like. So what we can do is we can say for all of the antibodies that we've characterized so far in the lab, what positions in the virus do those antibodies bind? So they bind to all sorts of different sites, but especially sites like 484. So you would expect that the virus might start off by getting mutations at site like 484. And so once the virus gets a mutation at site 484, you've knocked off the antibodies that bind at 484 and also antibodies that bind at other sort of similar regions like 490. So then you might say, all right, the next mutation the virus might get might be at a position like 417, where it's going to start knocking off the remaining antibodies. And then once that happens, there's other positions where future mutations might happen. So we think that this sort of visual calculation here can sort of be used to help understand how the effects of mutations at multiple positions in the virus might add up over the course of time. Okay, so just to end with some conclusions, one of the really kind of most longstanding challenges in evolutionary biology is to relate a change in the genotype to a change in the phenotype. And I talked about how even going back 55 years ago, evolutionary biologists have been worrying about doing that and worried by the fact that you can't just count up the number of mutations to understand the type of change you're going to get. And I obviously told you that we're still worried about that question today. For instance, we're trying to figure out what mutations in SARS-CoV-2 might mean for us. And in fact, I would say that understanding what mutations mean is increasingly the bottleneck in understanding virus evolution. So I sort of first started to study virus evolution in around 2009, which is when there was the 2009 swine flu. And people were able to sequence that, but it was still a relatively slow process. And now I've sort of lived through a second pandemic, which is SARS-CoV-2 pandemic, where now there are just millions or hundreds of thousands, over 1 million of sequences out there. And really sequencing the virus is becoming less and less the limitation. And now increasing the limitation is actually understanding what these mutations mean. And I hope I've showed you how we can at least start to make these big maps of the effects of mutations, like at these links I showed you here, which should enable people to interpret the effects of these mutations as they see them, so we can really understand and react to the virus's evolution, at least hopefully in close to real time. Finally, I'd like to end with some thanks. I'd like to thank Tyler and Ali in my lab who developed this system to measure the effects of mutations to the SARS-CoV-2 receptor binding domain and have done most of the really great work I talked about today. I'd also like to thank Rachel in my lab who did the really nice study of coronavirus 229E. Sarah, who's a former graduate student in my lab who built a lot of the interactive visualizations. And then Kate and Andrea, who developed a lot of the systems that we're using to actually sort of validate the effects of these mutations on virus. And then we've also been able to collaborate with a lot of other really great labs at the University of Washington and other places. And I'd like to thank my funding sources as well. And I'd be happy to answer any questions anybody has, and I will try to open the chat so I can start looking at them. Okay, so to go to the questions, so the first question from Kurt is, how sure are we that influenza transmits the way that we think it does? Yeah, so I mean, flu, to go to Kurt's question, flu definitely is a respiratory virus. I mean, it's transmitting at least partially through the respiratory route. I think what the question alludes to is that, you know, early in the SARS-CoV-2 pandemic, we were told a lot about, you know, surface contact and washing your hands, which is still a good thing to do. And then it became more and more clear the virus was transmitting a lot by airborne particles. I think for influenza, we actually at this point know less than SARS-CoV-2, but influenza transmits by some mix of surfaces, large droplets and aerosol droplets, but the relative importance of those different modes of transmission, I think, is still relatively unclear for flu. Another question, does this mean booster variant vaccinations for COVID won't need to be as frequent as flu? I think they probably won't need to be as frequent for COVID, or at least when they happen, they'll work better. So there's two problems with flu. One is that flu evolves rapidly, as I described. And the other one is that honestly, the flu vaccine isn't all that effective, even when we sort of get the viral evolution part right. It's just not that good of a vaccine in terms of the immune response it elicits. It's unclear exactly how fast SARS-CoV-2 will evolve relative to flu, but I'm hopeful that long-term it might be a little bit slower. But importantly, many of the vaccines against SARS-CoV-2, particularly the mRNA vaccines, work really well. And so you can imagine if you have better immunity to begin with, you can sort of take more hits from viral evolution before you need an update. So I think the COVID vaccine is going to be at least be better than the flu vaccine, even long-term. Susan asks, does the saturation mutagenesis bridge a little into gain of function, or do experimental safeguard systems? Yeah, so in our experiments, all of our experiments, we use only either the receptor binding domain as a protein, so that's not even a virus, or we use something that I didn't really get into here, but which is called a pseudovirus. So it's essentially a virus where we just have the spike protein and a few other genes, but are missing most of the rest of the virus. And so, yeah, so there's no danger, like nothing we're working on here is actually infectious in a human. So there's no concern of gain of function for that work. Can I share the link in the chat by any chance? It looks like it's been shared, but basically here's the link to the slides, and then everything else is linked there. Okay, sorry, I've kind of lost track of, okay. So Aaron asks, what do you mean when you say X company person developed this antibody? Yeah, that's a great question. So the monoclonal antibodies that are out there as treatments, so for instance, some of the first ones were developed by Regeneron or Eli Lilly. In those cases, basically what they did is they had pipelines to isolate antibodies from SARS-CoV-2 infected people. So from some of the first infected people, they got cells, B cells from those people. They cloned out a bunch of these antibodies, and then they used various criteria to decide which of these antibodies they thought would be good. And then they used sort of biotechnology to make a whole bunch of that antibody, and then it can be given to someone who's been infected with SARS-CoV-2. So that's sort of how a monoclonal antibody treatment works. And in general, for respiratory viruses, these monoclonal antibodies are quite effective if you're sort of given them early enough. That the problem that sort of limited the utility of monoclonal antibody treatments against SARS-CoV-2 is that a lot of the viral replication happens before you get sick. So the antibodies block viral replication. And you get infected with SARS-CoV-2 and you don't even know you're infected for 4 or 5 days. And so maybe by the time that someone's been given the monoclonal antibody, it might sort of be too late. And so this has been the biggest thing that's limited the effectiveness of those antibodies. I do think there's going to be an important niche for monoclonal antibodies long-term, though, in immunocompromised populations, including, for instance, cancer patients who are getting various immunomodulatory drugs who may not have a good natural immune response. And so there, you could imagine these antibodies could be given prophylactically, sort of before someone's infected, just to kind of protect them. And if the pricing of that works out, well, that could be a really important thing in immunocompromised populations. Okay, I'm going to get back to Kurt's questions, but since Kurt's had a lot of questions, I'm going to also get to a few other ones. So Stephen and Kara asked, is the difference in antibody focusing between influenza and measles more likely to be related to the number, structure of solvent-exposed residues on a targeted protein, or something intrinsic to the type of sequence that are recognized by antibodies? Basically, the short answer is, and I know it sounds kind of amazing, but we don't really know. So certainly antibodies typically bind to solvent-exposed things that are sort of out there on the surface. But beyond that, there's sort of a phenomena called immunodominance, where you show the immune system an antigen, for instance, a protein, and the immune system will often mostly hone in on one part of that protein. And it's often not exactly understood what determines that. And so this is one of the reasons that rational vaccine design is very hard. So one of the things that people have tried to do is design vaccines that would make the immune system target some different part of the virus, some part of the virus that it might seem better for the immune system to target. But it's pretty hard to instruct the immune system on exactly how it should bind to something. And people are definitely working on that, but it's been a real challenge. And this is why rational vaccine design has been difficult. And still, for reasons that we don't totally understand, there are some things like measles where basically these crude vaccines that were developed over a half century ago were great. And there's other viruses where even the most sophisticated, stuff that we can do now still doesn't really seem to be making the immune system do what we want it to do. Charles asks, how is ACE2 activity altered by virus binding, and is this variable in patients? So my understanding, and I'm not an expert on this, is that that probably the activity of ACE2 isn't that important in the pathogenesis of the virus. There's some question about whether ACE2 levels, like the expression level of ACE2, varies in meaningful ways as a function of age or other parameters that could affect disease severity. But I think, at least as far as I've been able to track in the literature, it's still a little bit unclear how much ACE2 expression is really a determining factor in the severity of disease or whether it's other factors. Shinrei T asks, is it possible to use computational modeling prediction to simulate different possibilities of how the antibody can bind to an antigen? Yeah, it definitely is. And people are definitely trying to do that, although it's still, and actually David Baker and Neil King, who are both professors at the University of Washington, are some of the best people in the world in doing that. And there's been a lot of progress made there, but it's still hard to do. Can we use a decoy ACE2 receptor as a prophylactic? Yes, that is something that is being tried. Whether that's going to be better than an antibody or not is sort of unclear to me. I think the antibodies are the ones that have made it into the clinic faster, but I think that probably has to do with reasons downstream from the molecular mechanism that I'm not as knowledgeable about. And so it's definitely a good idea. This is Ryan's question that people are pursuing. Barbara Dunn asked, if a virus has coexisted for humans with a long time, could it have evolved to be less quickly mutating compared with a newly introduced virus? Possibly. There's not really a lot of evidence for the mutation rate changing in a meaningful way for these viruses, although it's hard to measure mutation rates accurately. Certainly, sometimes the rate that the virus actually is evolving will change. So with SARS-CoV-2, there's two things going on right now. One is that the virus is getting mutations that are evading antibodies. And that's a process that will probably continue forever, essentially. But the other one is that when SARS-CoV-2 came into humans, it wasn't perfectly adapted to humans. So just transmitting even among people with no immunity, the virus was adequate at it, but not great at it. And the virus has gotten better at transmitting among humans. And I would expect at some point the evolution of increased transmissibility is going to plateau. A virus can't become infinitely transmissible. There's got to be some plateau. So I do think increased transmissibility will slow down eventually. Okay, so going back to Kurt's questions, does that mean we can use new technology like the mRNA vaccine to make a flu vaccine that works for more than a few months? I think Kurt answered the question very well, which is maybe, hopefully. I do think that we are going to see a lot of use of mRNA vaccines against a lot of other things. And, although I don't think we can say for sure, probably it's going to be better than the technology that's used right now for the flu vaccine. So I do think it's potential that we could see mRNA vaccines being useful against flu and other viruses as well. Kurt says, maybe Australian public health advice for flu is weird. Ours focuses on hand washing and covering coughs. It's not regarded as airborne like COVID and measles. I think that's accurate. I think flu is not as, measles is exceptionally transmissible. So transmissibility of a virus is measured by the R0, which is like if you have one infected person and they come in contact with other people who aren't infected, how many people on average should they transmit to? And the R0 for measles is something like 20. So measles is exceptionally transmissible. For flu, it's more like 2 to 2.5, so much less transmissible for measles. And I think for the new SARS-CoV-2 variants, it's getting in the range of like 5 to 6. So flu is less transmissible than COVID and measles. That's why all the social distancing that's been done around COVID has knocked down flu transmission a great deal. And probably that's because flu is less airborne transmissible than COVID, although I do think that there's good evidence that there is some airborne transmission of flu as well. And then, okay, I think I am more or less caught up on the questions in the chat. So if anybody has any more questions, feel free to unmute yourself and ask them or type them into the chat.
I have a question.
Mm-hmm.
One of the big problems in trying to design, for example, anti-malaria vaccine, but it's true for HIV as well, is that everybody's trying to do it with a single kind of immunogen, as exactly what's being done with the COVID too. And
yeah,
that's sort of asking for trouble. I mean, as you said in the course of your talking a while back, that some of these old vaccines, which are incredibly, sort of threw everything but the kitchen sink into the immunizing system. Polio is one good example. And in those cases, you find a much more robust in terms of evading evolution than when you have sort of one thing, in this case.
Yeah. So the question is basically like, should we be putting more than spike into the vaccine? So I can't really comment on malaria because I'm not that knowledgeable about malaria. And malaria, I think, is a very different end of the spectrum than these viruses, because obviously malaria is a larger organism that has a lot more proteins and targets in there. And I think its mechanisms of immune evasion are somewhat different. I mean, for SARS, so for most antiviral vaccines, certainly against acute viruses, so HIV is a chronic virus and it's a little bit different there. For acute viruses, the accepted correlate of protection of all the vaccines is neutralizing antibodies. That doesn't mean that that's a mechanism of protection, but it's at least the accepted correlate of protection. And you really only get neutralizing antibodies against the entry protein. That said, it's certainly known that for influenza, and it's probably true for SARS-CoV-2, that things like T cells can play an important role in regulating the severity of infection. So T cells are not going to provide someone with sterilizing immunity, so they're not going to keep you from getting infected. But once you get infected, they can do a lot to mitigate the severity of the disease. And in the case of influenza, actually, your point is a good one. There's two proteins on the surface of influenza, hemagglutinin and neuraminidase, and I think it's becoming increasingly accepted in the flu field that we should worry a little bit more about antibodies against neuraminidase as well. In the case of SARS-CoV-2, the spike is really the only reasonable target for neutralizing antibodies. That being said, probably T cells are going to play a significant role in mitigating disease severity. And so I think in that sense, it's important to think about, is the spike protein itself, which is a big protein, an adequate target for T cells, or would it be good to have additional T cell antigens in there? And I don't really have the answer to that.
Or even Novavax is, we're reading anyway, is trying to design one where you take take a look at some of your data and say, okay, these are the ones that we really need to worry about. So let's all in one immunization.
Yeah. Yeah, exactly. And I do think these sort of cocktail immunizations are really powerful. So it is a question, but I mean, I do have to say it does appear at this point that at least as far as neutralizing antibodies and probably protection go, the mRNA vaccines, so SARS-CoV-2, which are just the spike, are working as well as any of the vaccine formulations. Although you're also correct that, actually, now that I think about it, essentially all the vaccine formulations are predominantly spike. On the other hand, a live virus SARS-CoV-2 vaccine that's been inactivated, because SARS-CoV-2 is a high containment pathogen, at least until there are attenuated strains developed, the manufacturing of very large amounts of inactivated SARS-CoV-2 vaccine might have its own challenges.
Yes, indeed. So yeah, anyway, no, so the point just being is that the data you're generating are really useful and important. People are thinking the way I'm trying to get people to think sometimes when I...
Yeah. No, but I also think your point is well taken as well, which is that what we're measuring are biochemical correlates of antibody binding or immune protection, and it's unclear what are the actual mechanisms of protection in humans. I mean, we have some idea, but it's not totally known.
Yeah, but your data sets obviously are hugely important and useful.
Yeah.
People can use them in a variety of ways.
Hopefully they're useful. And let's see, Charles is asking, do low frequency errors in synthesizing RNA for vaccines contribute to antibody diversity in patients? I don't know the answer to that. My guess would be no, because my guess would be the error rate's fairly low, but I don't actually know the answer to that. I do know that there are companies that are exploring making sort of cocktail vaccines. So for instance, Moderna is now doing phase 1 trials with sort of a next generation booster vaccine. And one of those is just sort of the original strain of SARS-CoV-2 spike again. One is one of these variant strains and the other one is mixing the two together. And you can imagine this multiplexing of different variants that an mRNA vaccine could be effective. I think it's a little bit unclear, at least it's unclear to me, when you show the immune system a whole mix of different things, do you get kind of a broader response against all of them, or does it somehow make everything worse? I think that's unclear at this point, but I think it's potentially a promising area. Okay, so we're almost at, I think we are at 8, so we should probably, I don't want to make people sit here forever.
Thanks for the great talk, Jesse, and thanks to everyone for tuning in and staying overtime. And I hope a lot of you can come back next week for our final Wednesday evening at the Genome. We'll zoom out a bit and hear from Malia Fullerton about some big questions related to science and society, which should be very exciting. So have a good night, everybody.